let me introduce the names of the important enemies for us. So, classification of microbes. Classification of microorganisms. First one, viruses. Now guys, a peculiar feature about viruses is that they are neither living nor they are non-living. Okay, they are not organisms. The very first point you need to remember about viruses is that they are not organisms. They are, in other words, they are not cells. Okay, they are merely infectious particles. They are neither living nor they are non-living. Okay, and a virus cannot multiply itself unless and until it enters or hijacks a living cell. So an appropriate phrase, an appropriate descriptive phrase for viruses is that these are obligate intracellular infectious particles. Do not call them as cells. As I have mentioned, they are neither living nor they are non-living. So they are obligate intracellular infectious particles. There's a reason why I have picked up viruses as the very first subheading in this heading of classification of microbes. You know why? Now guys, what I want all of you to pick up from this cartoon which I have drawn, what is this? You can see Tata car, this is the nano car. Okay, why is this car named as nano? Because nano means what? Nano means small. And viruses are the smallest agents which can infect us. How small they are? They are so small that their size is measured in nanometers. Their size is measured in nanometers. And 1 nanometer is equal to 10 raised to the power minus 9 meters. Okay, guys, it is easy to remember. N for nano, N for 9. N for nano, N for 9. Okay, so these are obligate intracellular infectious particles. They are very small. How small? Their size is measured in nanometers. Okay, and the other thing which you need to remember about viruses are they have got either DNA or RNA, never both. Never both. Okay, you can remember it like this. These poor guys are so small that they do not have enough space to accommodate both DNA and RNA. Okay, this is extremely important point. They have got either DNA or RNA but never both. And guys, this in fact is the basis of classifying viruses as DNA viruses or RNA viruses. And how viruses multiply? They do not multiply by binary fission or mitosis or meiosis. Instead, there's a very unique method by which viruses multiply or replicate, wherein when one virus enters a cell, it will manufacture hundreds and thousands of daughter viruses, which is amazing in its own self, and we'll come to details of how that happens during virology modules. For now, you can just remember that viruses multiply by what is known as viral infective cycle. They multiply by what is known as viral infective cycle. Okay guys, so these are the first enemies which you need to learn about, the different kinds of viruses which can make humans sick. Okay, now coming to the second category of the villains or the enemies, we have got bacteria. Okay, now these are the second enemies which you need to know about. Now guys, at the onset only I would give you all a suggestion. while taking down these notes, as I am using different colors and highlighters, I would highly recommend the use of different colors and highlighters by all of you also, because it will help you a lot in remembering stuff once you have got colorful notes with you. Okay, so B for bacteria. Remember, B for bigger. These agents are bigger than viruses. B for bacteria, B for bigger. How much big? Their size is measured in microns or micrometers. Microns or micrometers, and 1 micron is equal to 10 raised to the power minus 6 meters. Remember, 1 nanometer is equal to 10 raised to the power minus 9 meters, and 1 micron is equal to 10 raised to the power minus 6 meters. Okay. So guys, as you are watching this video module, I would want all of you to remember the stuff along with me only. Okay. So we'll not only make notes during this video module, but we'll memorize also. Okay. So remember, B for bacteria, B for bigger than viruses, then B for bacteria and B for both. Bacteria have got both DNA and RNA, unlike viruses. So B for bigger, B for bacteria, B for both DNA and RNA. And the other thing which I want all of you to remember is, again, B for bacteria, B for binary fission. Binary fission is the method of bacterial replication or multiplication, one and the same thing. Bacteria have been classified as what? Now I am going to use the short form B for bacteria. Okay. Remember, one single organism, if we talk about one single organism, is referred to as bacterium. And if we have got multiple bacteria, we call them as bacteria. Okay, so strictly speaking, the term bacterium means one single organism and bacteria means multiple ones. Okay, so I am going to use the letter B henceforth. Now remember, bacteria are unicellular or single-celled organisms. They are single-celled organisms, and remember, they are prokaryotes. Bacteria are prokaryotes. Now let me dissect this term for you. Now guys, prokaryotes, pro means primitive or old, and the term karyon means what? Nucleus. Karyon means nucleus. Do bacteria have a nucleus? No. Bacteria do not have a nucleus. Instead, they have got what is known as nucleoid. Now let us understand that prokaryotes have got no nucleus. Now what do I mean by the term nucleoid? Imagine this orange box which I have drawn. Imagine this is a bacterium. Okay, one organism, a single bacterium this is. Okay, happily smiling bacterium. Now what am I drawing in yellow? Now imagine, see, I have drawn two sort of circular wavy structures. Okay, these are looking like two Maggi noodles. Now imagine these two Maggi noodle strands are intertwined with each other. Okay, so what I want you all to remember is, even though I have got two strands, the two strands are intertwined or nicely wounded into each other. What is this representative of? This is nothing but the bacterial genome. So bacterial genome is what? It is nothing but a single circular double-stranded, single circular double-stranded. The short form DS is for double-stranded. It's a single circular double-stranded DNA molecule. DNA molecule. Okay, so this is nothing but a bacterial genome. Okay, Now, bacteria have got a single chromosome which is freely lying in the cytoplasm, okay, and there is therefore no need of a specialized structure called as nucleus which is there in eukaryotes, okay. We have got just a single circular double-stranded DNA molecule lying freely in the cytoplasm of a bacterial cell, and that area in the cytoplasm where this bacterial genome is located, that area is referred to as nucleoid. Remember, nucleoid word is just a descriptive term for the space of the cytoplasm occupied by the bacterial genome. It is not a specialized structure per se. So, nucleoid is nothing but the space or volume of the cytoplasm occupied by the bacterial genome. Okay, occupied by the bacterial genome. Not to be confused with nucleus at all. Nucleus is a specialized structure. Nucleoid is just a descriptive term. It does not mean any structure. Okay, this is very important. The definition of bacterial genome. Then next, what you need to know about bacteria. Bacteria have a cell wall made up of, now I am going to tell you a very fancy word which I am going to dissect, talk about bacterial cell structure and anatomy. For now, just note down this fancy word, peptidoglycan. So, bacteria have got a cell wall made up of peptidoglycan, also known as mucopeptide or muramic acid or murine cell wall or murine cell wall. Now guys, this fancy word peptidoglycan or mucopeptide or muramic acid or murine, you are not going to hear about this word anywhere else. It is a term used only for bacteria and for no other organism. Okay, so we'll come to this later on. Now next, now guys, if you keep aside viruses which are neither living nor non-living, and you just take into consideration the other villains, for instance bacteria, the fungi and the parasites which include protozoa and helminths, all these are living organisms unlike the viruses. Okay, bacteria, parasites, fungi, right? Now out of all these living organisms, I personally look upon bacteria as the odd fellows. You know why? Because bacteria have got prokaryotic ribosomes which are 70S made up of 2 subunits, 50S plus 30S. Okay, so remember 7, 5 and 3. What are these? These are odd numbers. Okay, and bacteria are prokaryotes. They are the odd fellows out. If you compare bacteria with the other villains which are eukaryotes, for instance the fungi and the parasites. Okay, so all those eukaryotes are in a separate category of eukaryotic organisms, and they have got eukaryotic ribosomes which are even numbered ribosomes, right, which I'll come to later on. Bacteria are the odd fellows out. They are the only prokaryotes which you need to know about, and because they are odd, remember they have got odd numbered ribosomes, that is 70S made up of 2 subunits, 50S and 30S. So remember 7, 5 and 3. So this is about bacteria. Now the third kind of villains which you need to be aware about are fungi and protozoa. Now protozoa are a kind of or a type of parasites. Okay, I am clubbing them together, fungi and protozoa, in category number 3. Now remember, the very first feature which you need to know about these is they are eukaryotes. Okay, they are eukaryotes. Eu means true and karyon means nucleus. Now nucleus is a specialized structure. Okay, it is a specialized structure which has what? Which has the chromosomes or the eukaryotic genome, And remember, in eukaryotes, there are multiple chromosomes which are linear. So, there are multiple linear chromosomes which are located in a specialized structure called as the nucleus. Now guys, what you can see, okay, somebody is talking to you. What is this? This is a locker. Okay, hey folks, I safeguard Bapuji, in other words, money. Okay, people who have got a lot of money, where do they keep it? In the locker, in their houses or in the bank. Okay, now analogous to this locker, we have got what? We have got the nucleus in eukaryotes. Okay, there's a talk going on between the locker and the nucleus, and the debate is about who is more important. Okay, so both have got their own ego issues, the nucleus as well as the locker, and both of them are standing their ground. As the locker is saying, I safeguard Bapuji, money is very important, and the nucleus is saying, well, I'm more important, I safeguard the genes. Okay, so you can understand now, bacteria have got just, bacteria are poor fellows, they have got just a single circular double stranded DNA molecule which is freely lying in the cytoplasm. There is no need for the presence of a locker in the form of nucleus in case of bacteria. Now eukaryotes on the other hand, they have got multiple, eukaryotic organisms are rich in the sense that they have got multiple chromosomes which are linear, and they have to accommodate all these chromosomes in a specialized structure which God has given to them in the form of the locker of nucleus. Okay, so now it's understandable why nucleus is important in case of eukaryotes. Okay, now so the common thing between fungi and protozoa is that both are eukaryotes, they have got a true nucleus. Now let us see some differences. If you write protozoa and you highlight the O's, there are 3 O's. Now these O's look like what? They look like 0, and from 0 I want all of you to remember that protozoa are unicellular or unicelled organisms. They are unicellular. Fungi, on the other hand, could be unicellular or multicellular. Alright, so we have got 2 kinds of fungi. Some of them are unicellular, made up of just one cell, whereas some fungi are multicellular. So this is one point of difference between protozoa and fungi. Now I am going to use the short form P for protozoa and F for fungi. Now fungi, what is the size of fungi? Remember the range 5 to 15 microns, easy to remember. F for fungi, F for 5, F for 15. Okay. Now protozoa are a little bigger. They roughly measure between 10 to 20 microns. This is just a range. Of course, there could be a little exceptions, but most of the fungi and protozoa fall in this range of the size. So remember this much. And next important point, as I told a few minutes back, eukaryotic ribosomes are, remember the even numbers, 80S made up of 60S plus 40S. So remember 8, 6, 4. These are even numbers. E for even numbers, E for eukaryotes. Okay. So this is how eukaryotic ribosomes differ from prokaryotic ribosomes. Now guys, this knowledge is important not just from the point of view of knowing about these microorganisms, but we make use of this knowledge of the different kind of ribosomal existence in bacteria and the eukaryotes, because we can devise or formulate drugs which specifically attack or target the prokaryotic ribosomes. Remember, we the humans, we obviously are eukaryotes. Okay, so if suppose we devise an antimicrobial drug which attacks the eukaryotic ribosomes, if suppose we want to attack some fungus or some parasite, a side effect of such a drug would be it will also attack our ribosomes. All right. So if we know about the existence of different kind of ribosomes, we can specifically come up with some drugs with some modifications so that they either harm us less with lesser side effects, or we can come up with a separate category of drugs altogether which target only the bacterial or the prokaryotic ribosomes. Okay, so that is how all this knowledge comes handy to us. Now, as far as multiplication is or replication is concerned, asexual multiplication and sexual multiplication both are seen in case of eukaryotes. Asexual mode is mitosis and sexual mode is meiosis. All right. Now, guys, please do not confuse binary fission with mitosis. Binary fission, where did I tell you? B for binary fission, B for bacteria. Okay, so binary fission is the mode of multiplication in bacteria. It is not at all synonymous with mitosis. Remember, in mitosis, as we have learned in +2 classes in school also, a mitotic spindle is formed. Why you think this is required? Because there are multiple chromosomes in eukaryotes, and during cell division, what happens? Every chromosome produces its Xerox copy. Okay, and while division of a cell into two daughter cells during mitosis, it's very important that both the daughter cells get one copy each of a particular type of chromosome. For instance, suppose there is chromosome number 2. Okay, so both the daughter cells should be having one copy of chromosome number 2. Both the copies of chromosome 2 should not be given to one single daughter cell. And to make sure that all this happens properly and appropriately and safely, mitotic spindle is formed. All right, but no such requirement is there in case of bacterial replication, where the bacteria just have one single chromosome. So in case of binary fission, there will just be replication or Xerox copy formation of the bacterial chromosome, followed by the cytoplasmic division, and we will have two bacterial cells out of a single parent cell. All right, so remember, binary fission and mitosis are not at all synonymous. So this is about the third category of the villains, fungi and protozoa. And lastly, we have got a very special parasites called as helminths. Now guys, these are also eukaryotes, but why did I not consider them in category 3, where I included fungi and protozoa? Now guys, these are special parasites. Even though these are also eukaryotes like protozoa and fungi, I still am giving them more importance by designating a separate category altogether. The reason is they are not microbes. Instead, they belong to animal kingdom, and their adults are big enough to be seen with naked eye. Then why are we studying them in microbiology if they are not microbes? Remember, there are certain structures pertaining to helminths like helminthic eggs, helminthic larvae. They need the assistance or the aid or the help of a microscope to be seen. Okay, that is why we study them in microbiology, even though the adult forms are big enough to be seen with naked eye. Alright, so these are the important categories of the villains which infect us. Remember, the smallest ones, nanocar, viruses, then bigger than them, bacteria, then which are prokaryotes. Then the third category is fungi and protozoa, both of which are eukaryotes. Okay, and lastly we have got helminths. Okay, so these are the conventional villains. Now I am gonna tell you two more names of unconventional villains or the unconventional infectious agents. Okay, now guys, life also, we sometimes befriend somebody, but later on that person turns out to be actually an enemy for us, or not the well-wisher as was perceived by us before. Similarly, there are some unconventional infectious agents. Okay, unconventional infectious agents I personally refer to them as, VIPs, VIP microorganisms, the unconventional ones. Remember, V, I and P. V actually stands for viroids. I stands for nothing, that's why I have written small I, and P stands for prions. P stands for prions. So remember this mnemonic for the unconventional agents, V, I and P. Now what are viroids? Now first point to be remembered about viroids is that they do not infect humans. They cause merely plant diseases. They cause plant diseases, and what exactly are these? These are covalently closed single RNA molecules. Now, what do I mean by this? Imagine if this is an RNA molecule. Okay. Now, this RNA molecule has some complementary bases within it. As a result of those complementary bases, what will happen? It will circularize like this because of bond formation between the complementary bases. So, this is what is meant by single RNA molecules which are covalently closed by this. Good news about viroids is they do not infect humans. So far they have not been found to cause any human disease, they cause only plant diseases. Now, on the other hand, prions. Prions are what? Prions are the, at the opposite end of the spectrum if we compare them with viroids, in the sense that they are, remember, PR, so these are proteinaceous infectious agents. So, PRINS, these are proteinaceous infectious particles. They are nothing but proteins. Proteinaceous infectious particles. They don't have any nucleic acid. They are devoid of nucleic acid. Okay, so this is in direct contrast with the viroids, which are nothing but nucleic acids. What kind of nucleic acid? They are RNA molecules. Prions, on the other hand, are devoid of any kind of nucleic acid. Instead, they are protein particles. Remember, P for prions, P for proteins. Do they cause human disease? Yes. They cause rare human diseases, which are referred to as, they cause human and also animal diseases, and the human diseases which they cause, they are referred to as spongiform encephalopathies, simply referred to as prion diseases, and they were discovered by Stanley Prusner, who also was awarded a Nobel Prize for the same. Okay. So, remember these unconventional infectious agents, the viroids and prions.